Let's be real, remote learning isn't just school from home. It's trying to stay awake during the 9 a.m. lecture while your bed is exactly 2 feet away, your Wi-Fi is fighting for its life, and you haven't seen another human classmate in 3 days. If you're trying to ace your classes without losing your mind from your bedroom, you're in the right place. CBSE 12, Biology Unit V, Reproduction, focusing on Chapter 1, Sexual Reproduction in Flowering Plants, along with an overview of the introductory sections. 1. Unit Overview and Introduction, the Biology of Reproduction. Individual organisms inevitably die, but species persist for millions of years through reproduction. Sexual reproduction specifically creates new genetic variants, enhancing survival advantages. The unit covers reproductive processes in flowering plants and humans, as well as reproductive health. Tribute to Panchanan Maheshwari, 1904–1966, a world-renowned Indian botanist who specialized in plant morphology and embryology. He popularized the use of embryological traits in taxonomy, advanced artificial cultures of immature embryos, and achieved worldwide acclaim for test tube fertilization and intra-ovarian pollination. He also led the creation of the first NCERT biology textbooks for schools in 1964. 2. The flower. The organ of angiosperms, human significance. Flowers possess aesthetic, ornamental, social, religious, and cultural value, acting as symbols for human feelings like love, happiness, and grief. Biological marvel. To biologists, flowers are morphological and embryological marvels functioning exclusively as the structural sites for sexual reproduction. 3. Pre-fertilization. Structures and events before a flower blooms. Hormonal and structural changes differentiate the floral primordium into inflorescences, buds, and finally flowers containing male reproductive organs, androecium, and female reproductive organs, gynoecium. A. The male reproductive organs, stamen, microsporogenesis, and pollen grain, stamen structure. Composed of a long slender stalk called the filament, attached proximally to the thalamus or petal, and a terminal, usually bilobed structure called the anther. Anther anatomy. A typical angiosperm anther is bilobed and dithecous, each lobe containing 2 theca. Structurally, it is a 4-sided, tetragonal, organ hosting 4 microsporangia located at the corners. Microsporangium wall layers. Surrounded by 4 distinct layers: epidermis, endothecium, and middle layers. The outer 3 layers that protect the anther and assist in its opening, dehiscence, to discharge pollen. Tapetum. The innermost, nutrient-dense layer featuring dense cytoplasm and often multiple nuclei, which nourishes developing pollen grains. Microsporogenesis. Young microsporangia contain a central mass of homogeneous cells called sporogenous tissue. Each cell acts as a pollen mother cell, PMC, and undergoes meiotic division to generate a cluster of 4 haploid cells known as a microspore tetrad. As the anther matures and dehydrates, these separate into individual pollen grains. Pollen grain, male gametophyte, typically spherical, 25 to 50 micrometers in diameter, with a 2-layered wall. Exene, outer layer, made of spiropylenin, an incredibly resistant organic material capable of withstanding extreme heat, strong acids, and alkalis. It features small openings called germ pores where spiropylenin is absent. Intine, inner layer, a thin, continuous layer composed of cellulose and pectin. Cellular development. A mature pollen grain contains a large vegetative cell, abundant food reserves, irregular nucleus, and a small, spindle-shaped generative cell floating in its cytoplasm. In over 60% of angiosperms, pollen is shed at this 2-celled stage. In others, the generative cell divides mitotically into 2 male gametes before shedding, 3-celled stage. Impact and uses. Pollen can cause severe respiratory allergies, e.g., parthenium, carrot grass. Conversely, pollen tablets and syrups are consumed as nutrient food supplements to boost athletic performance. O viability. Viability ranges from 30 minutes, rice, wheat, to months, Rosaceae, Solanaceae. Pollen can be preserved for years in liquid nitrogen, $196 carat, circ, text, C, $, inside pollen banks for crop breeding. B. The female reproductive organs, pistil, megasporangium, and embryo sac. Genetium structure. The female part can be monocellary, one pistil, or multicarpellary, multiple pistils. Multiple pistils can be fused together, syncarpous, or free, apocarpous. Pistil anatomy. Consists of a stigma, landing platform for pollen, style, elongated structural tube, and ovary, basal bulged part containing an ovarian cavity, locule and a placenta. The megasporangium, ovule, attached to the placenta by a stalk called the funicle. The junction where the ovule body connects to the funicle is the hilum. The ovule consists of O protective envelopes called integuments that encase a food-storing cell mass called the nucellus. OA small tip opening called the micropyle, opposite the basal chalaza pole. Megasporogenesis and female gametophyte. A single megaspore mother cell, MMC, in the nucellus undergoes meiosis to produce 4 megaspores. In most plants, 3 degenerate, and only 1 functional megaspore undergoes 3 free nuclear meiotic divisions to create the female gametophyte, embryo sac. This is termed monosporic development. Mature embryo sac organization. A typical mature angiosperm embryo sac is 8 nucleate but 7-celled. Arranged as follows. O egg apparatus, micropylar end, 2 synergids, featuring a filiform apparatus to guide the pollen tube, and 1 egg cell. O antipodals, chalazole end, 3 cells. O central cell. A single large cell located in the middle containing 2 polar nuclei. Look, nobody ever hands you a manual on how to actually be a successful remote student. It takes serious discipline, and a lot of caffeine. So today, we are skipping the generic advice and breaking down the exact, no BS strategies you need to stay productive, crush your deadlines, and actually reclaim your free time. 4. Pollination mechanisms. Because male and female gametes are non-motile, flowering plants rely on external agents to transfer pollen from the anther to the stigma. A. Kinds of pollination. 1. Autogamy, self-pollination, transfer of pollen within the same flower. Some plants, e.g., viola, oxalis, camelina, bear 2 types of flowers. O. Chasmogamous, standard open flowers with exposed reproductive organs. O. Cleistogamous, flowers that never open, forcing absolute autogamy and ensuring seed set even without external pollinators. 2. Geitonogamy, transfer of pollen to another flower on the same plant, functionally cross-pollination via an agent, but genetically identical to autogamy. 3. Xenogamy, cross-pollination, transfer of pollen to a flower on a different plant, introducing genetic variation. B. Agents of pollination, abiotic agents, wind and water. Wind-pollinated plants produce massive amounts of light, non-sticky pollen, alongside well-exposed stamens and large, feathery stigmas, e.g., corn cobs, grasses. Water pollination is rare, mostly monocotyledons like Vallisneria, Hydrilla, and Zostera. Abiotic flowers lack bright colors and nectar. Biotic agents. Animals and insects fulfill the majority of plant pollination. Certain relationships are obligate symbiotic linkages, such as a specific moth species and the yucca plant, which depend completely on each other to fulfill their life cycles. Note: organisms that steal nectar, pollen without causing pollination are labeled pollen, nectar robbers. See outbreeding devices and pollen-pistil interaction. Outbreeding devices. To prevent inbreeding depression caused by constant self-pollination, Plants use mechanisms like unsynchronized pollen release, stigma receptivity, physical displacement of organs, self-incompatibility, genetic rejection of self-pollen, and unisexual flowers, dioecious. Pollen-pistil dialogue. The pistil performs a continuous chemical dialogue with landing pollen to determine compatibility. Compatible pollen germinates a pollen tube via a germ pore. The tube grows through the style into the ovary, tracking signals from the synergid's filiform apparatus to enter the embryo sac. Artificial hybridization. Used by plant breeders to create superior crop varieties. It relies on emasculation, removing anthers from bisexual buds using forceps, and bagging, covering the stigma with butter paper, to safeguard the female flower from unwanted pollen contamination. 5. Double fertilization. Once the pollen tube breaches a synergid, it discharges 2 male gametes. 1. Xenogamy. One male gamete fuses with the haploid egg cell nucleus, yielding a diploid, $2 zygote. 2. Triple fusion. The second male gamete moves to the central cell, fusing with the 2 polar nuclei to establish a triploid, $3 primary endosperm nucleus, pen. Because both xenogamy and triple fusion happen within the same embryo sac, the event is uniquely named double fertilization. The central cell transforms into the primary endosperm cell, PEC, to generate the endosperm, while the zygote grows into the embryo. 6. Post-fertilization. Structures and events. A. Endosperm development. Endosperm growth always precedes embryo development to ensure a constant supply of nutrition. In free nuclear endosperm, the most common type, the pen undergoes consecutive meiotic divisions without immediate cell wall formation, e.g., coconut water. It later undergoes cell wall formation to become cellular endosperm, e.g., white coconut kernel. Albuminous seeds retain part of the endosperm at maturity, e.g., wheat, maize, castor, coconut, whereas non-albuminous seeds completely consume it during embryo growth, e.g., pea, groundnut, beans. Persistent remnants of the nucellus in a mature seed are called perisperm, e.g., black pepper, beet. B. Embryo development, embryogeny. The zygote sits at the micropylar end and transitions through preembryo, globular, heart-shaped, and mature embryo stages. Dicotyledonous embryo consists of an embryonal axis and 2 cotyledons. The section above the cotyledons is the epicotyl, ends at the plumule, shoot tip, and the cylindrical section below is the hypocotyl, ends at the radical, root tip, shielded by a root cap. Monocotyledonous embryo possesses only one cotyledon, called a scutellum in the grass family, placed laterally. Its radical and root cap are enclosed in a sheath called the coleorhiza, while its shoot apex and leaf primordia are protected inside a hollow foliar structure called the coleoptile. C. Seed and fruit development, seeds. The final product of sexual reproduction, a fertilized ovule. Integuments harden into tough protective seed coats, leaving the micropyle as a small pore for oxygen and water intake during germination. As it matures, it dries, 10 to 15% moisture content, and the embryo may enter a metabolic standstill called dormancy until favorable conditions return. Fruits. The ovary wall transforms into the fruit wall, or pericarp. O. True fruits. Develop strictly from the ovary. O. False fruits. Floral structures other than the ovary contribute to the fruit. For example, the thalamus forms the fleshy part of apples, strawberries, and cashews. If you made it this far, you're officially part of the remote student grind. Do me a quick favor. Smash that like button if this helped you out. Share this with a classmate who is currently drowning in online assignments. And hit subscribe so you don't miss out on our next video dropping day of the week, e.g., this Thursday, where we're tackling next video topic, e.g., how to build a study schedule that actually works. Until next time, stay focused, close those extra tabs, and I'll see you in the next video.